So, good morning. It's my great pleasure to introduce to you Eva Morava, who is visiting us from Nijmegen, the Netherlands. Eva got her MD, about 1990 or so. I shouldn't say dates, right? She's extremely young, but she has completed training in medicine as well as neonatology and genetics, in which she's both board certified. She's an extremely productive young clinician with respect to metabolic diseases in particular. And today she's going to talk to us about the congenital disorders of glycosylation. I'll just tell you we're taping, and we also have a few people from around the region who are joining us for this, but hopefully that won't disturb. So, Eva, thank you so much for coming.
Good morning. It's an honor to talk to you today. It's a little bit scary how far you're sitting. Like there are 10 people and they are sitting in an auditorium. So I will try to climb closer to you, but sometimes I have to take care of my slides, so I think I'll exercise a lot today. I just heard about you guys that you are excellent in biology and you probably know all this, but I thought I'll start with some basics regarding glycosylation. And of course you'd think glycosylation, why glycosylation? Why is it so important? But actually it is important because the post-translational modification is so essential for our body, for our function, that almost 50% of our functional proteins are modified and glycosylated. We have several very important functions which we cannot have without proper glycosylation. So the first and the most important is protein folding. You can have the best protein ever. If it's not folded right, it would never work. So it's important for stability, for soluble. I cannot even pronounce it. But of course, a defense mechanism not to be broken down. It is important for the surface, for the immunological recognition. And many of the glycosylated proteins play an essential role in coagulation, cell-cell interactions, and also in developmental pathways and signal transduction. So you already see that my lecture is the most important lecture from this semester, because this is what you really have to know. So a little bit in general, we are talking about two major pathways of glycosylation. And one is the so-called N-linked glycosylation, and the other one is the O-linked glycosylation. And it's very easy to understand because it's actually just how the antenna, a glycan antenna, is linked to the protein. And when it's linked with an N group to an asparagine, then we call N-linked glycosylation. And when it's linked with a hydroxy group to a serine of 3 and 9, then we call it O-glycosylation. This seems to be irrelevant now, but later it will get important. So listen to each word. Should I speak up? Even more speak up?
Yeah, you're fine.
Oh man. So, just to emphasize my own importance, I show you that glycosylation is important everywhere. Without proper protein glycosylation, you cannot have a normal cardiac function, and you get a cardiomyopathy. If you don't have proper glycosylation, your enteral absorption is abnormal because your gut is just covered with these glycan antennas, and they are helping in absorption, but also protecting your gut from infections.
Sorry, we didn't bring you in.
Oh man.
I'll get you just a second.
This technical thing is killing me. The kidneys, they really need normal glycosylation, otherwise you would lose all that stuff through the tubuli. Oh man, now it gets complicated. Most of the functional proteins of our muscles are glycosylated proteins as well. So when you're missing them, you get a severe muscle dystrophy. Most of our important hormones are glycosylated. It just gets better and better. So if you don't have that, you will have an endocrine disorder. Your brain, when it's developing, it is essential that these glycan antennas are on the cell surface. Otherwise, the cells wouldn't know where to migrate, and they just stay at a place where they cannot properly function. And coagulation. So it's, you see, you know now everything. When you don't have proper glycosylation, you're completely sick. You have no chance for survival. Still, it's amazing, but we have patients with systemic glycosylation disorders. Each cell of their body is missing the proper glycan antenna, and they are still alive. They have a very severe disease. We call this disease congenital disorders of glycosylation, or CUG syndrome. This is not only a developmental abnormality, but also a chronic metabolic disease. And you can assume the symptoms, because I just told you that all these organs, they're using glycosylation. So you will see, when you see a patient, that they show all these features, from hormonal problems to cardiomyopathy, from malabsorption, chronic diarrhea, to hypothyroidism. They have all these problems, but they have something really particular. You can recognize this syndrome just by looking at the patient. So I wonder if you see any particular when you look at this young lady here. Do you see the fat distribution on her arm? It looks like when you're losing weight or you're getting old, you know, it's a little bit hanging there. So they are born with this strange fat distribution, and you can see it on their arms, on their chests, and even more on their hips. They are looking like females in their early 40s, not like me, like in general, you know, when they're developing those nice hips. So they do have the fat deposition already after birth, and when you do a physical exam, you recognize these strange abnormalities. They also have strabismus. If you look at all these 4 patients have the eye problem, and they have quite a peculiar face. So many times you can just diagnose this syndrome based on their physical features. And if you really go more deep into that, you see what I told you is all right. They cannot feed normally. They cannot grow normally. They are hypotonic because their muscle glycoproteins are also abnormally formed. They cannot develop properly. They have an ataxia. I just told you that for proper brain development, you really need that signal, that glycan antenna on the cell surface, to tell the cells to migrate to the right place. So the vermis, so the cerebellar vermis in these patients is almost always underdeveloped. So they get also a movement problem, they get ataxia. Also, they have the endocrine abnormalities, abnormal liver function. Most of the liver proteins are glycosylated, and all functional liver proteins are glycosylated. So you can imagine that from alpha-1 antitrypsin to transferrin, they are all abnormal and not functioning right. So it's a wonder that they are living. Here what you see is the hypotonia. If you're moving a baby from this age, they're supposed to move their body and not just hanging like a puppet. Here you see those blue spots after delivery. The baby had so much bleeding, subcutaneous bleeding, that was completely blue. And here you see a patient who's tube fed because of this abnormal gut and abnormal absorption. Okay, so it's a miracle that they are alive, but it's even more a miracle that they get adult. Do I need a pen?
It's a pointer.
It's a pointer.
Oh, it's a pointer. I thought I need a pen. Somebody wants to have an autograph. I'm very important. So listen to that. These are patients of mine, and they were so sick when they were young. But this one is already 33 and has a twin sister, and she's also 33. No wonder, happy are twins. But they are alive, and they can walk, and they can talk. This patient cannot talk because of the cerebellar problem, but he learned to communicate with communication device. And this is also a patient who cannot talk, but uses a sign language, and is pretty happy. They are not frequently ill. They are, in general, very cheerful patients and very easy to take care for. What's happening in the adult life, it's not so spectacular. Usually they stay stable. If they get to the adult life, then usually they don't have too many new symptoms, but they can develop a symptom, retinitis pigmentosa. I'm not sure if you heard about that. That's a sort of pigmentary abnormality of your retina, and you lose your sight through that. So that's pretty bad. But that's happening only after the age of 30, 40. So this is a so-called CDG syndrome. And we were thinking that this is quite frequent, but the more I screen for, the more patients I find. And currently, we are taking care of 70 patients at our center. That's a lot.
Yeah. What's the status in general of this disease?
What is the frequency? Nobody knows because there's not really, this disorder is only discovered 15 years ago, so we are still collecting patients and knowledge. There are also some patients who are quite mild, and I have a patient who is 22 and is going to college, and the only symptom she has is titubation. It's a sort of tremor-like movement of her head, and she's not walking very stable, and she has to hold on sometimes, but intellectually and otherwise she's a lovely lady, and she has a boyfriend. So she has a full life. But this is the reason why sometimes we maybe underdiagnose the patients, because I wouldn't have tested this patient, only it was an accident, like most great discoveries. Somebody sent for the, not for the right test, and we screened her.
Are you screened?
Could you repeat the questions?
I will tell you.
Eva,
Right?
repeat the question.
Oh, I'm sorry. He asked how do we screen, but I will tell you just in a minute. But you wanted to ask something.
With the variety of the intensity of the disorder that you've shown, is it such that all the proteins don't have this problem of being not properly glycosylated? I mean, is it either all the cells get it or none of the cells, or that's why you see this variety of it? That's just some. I'm not understanding why you have such. It's not all or nothing.
But you are smarter than I am, and I don't understand either. The question was, how is it possible that it's so variable when you have like a systemic disorder? All your cells are involved, all your proteins are involved, and if you do test the proteins one by one, then you find the anomaly, but the clinical expression varies pretty impressively. I mean, I have a couple of theories how can it occur. And of course, there is a genetic background. Sometimes, depending on the mutation, those patients who are carrying nonsense mutations are not viable. And compound heterozygotes carrying a nonsense and a missense mutations are quite severe. And I mean, this is in general, it is true. Still, sometimes you don't understand. But I will talk about this a little bit later because I have so much to tell you. Maybe I cannot really go there.
Speak loud, don't move so much, and talk to that microphone.
Is this normal? I mean, I'm giving a talk to you guys, and I should speak louder and talk to the camera. But then you should get my slides going. But you don't see the slides anymore because of my slender body. So what I wanted to show you, that this variability makes my life difficult, because sometimes you look at all these patients and where's the thing? I mean, they look just fine. I won't say that they're particularly dysmorphic. Some of them are really just great and cute. So how to diagnose this inborn error? I have a slave now.
For life.
For life. So just get into the biochemistry a little bit, and then maybe I can give you some answers later on. This is really a complicated pathway. So we won't really go into all these details, each step of the pathway. But in general, glycosylation starts in the endoplasmatic reticulum, and then we call that type 1 CDG defect. And the glycosylation goes through the Golgi, and it's a ripening process. So there is a second step where you're doing not only N-linked but O-linked glycosylation. And finally, you go into the secretory phase when these ready proteins are secreted to the right place. So this is the thing. If you're really smart, you just memorize this pathway. Just the next slide, please. Look at how many different disorders are already known. Different subtypes of congenital disorder of glycosylation. These are the N-linked, so the endoplasmatic reticulum linked glycosylation types, and these are the O-linked. So it's about 36 different types we discovered in the last 15 years, and they are all different. So how would you diagnose? How would you find such a disorder? So you would start with the antennas. You go for the antennas. You want to learn what's wrong with the antennas. And then you think, are the antennas N-linked or O-linked? Because then I can look for certain enzymes which are producing the antennas. And then there is a barcode. You just screen all these patterns, and then you read the barcode and say, this is type 1, this is type 2. So this would be a wonderful thing if you could do like this. But it's not inconceivable that we can do it like this. Because of course, when you're building an antenna, you have to put the certain molecules like mannose and glucose and sialic acid one after the other, and then you have to have a carrier, and the carrier has to carry these antennas to the site where they undergo further processing, and finally somebody would put the antenna on the protein. So we only have to study how this processing is going on, and then we are in business. So DC, here's your answer. How do we screen? Very easily. We only need 1 drop of blood, actually just 1 drop of serum. And we use that serum because there are all these proteins in it, which are all glycosylated. So we just use 1 antibody, and we know that transferrin is a glycosylated protein. So we use transferrin. We could use alpha-1 antitrypsin or many others, but this is our favorite. So let's say this is our protein, and we have the antennas on it. They have sialic acid at the end. So this is mannose, this is glucose, this is sialic acid. Sialic acid is a negatively charged happy ending. When it's there, our molecule is negative. So if you have a gel, And you try to do isoelectric focusing. Did you learn about isoelectric focusing? And you are nodding. So you have a charge, and you try to get those proteins going on your gel and finding their isoelectric focusing point, and there they remain. And in a normal individual, in a control individual, you get just one spot because all these antennas are glycosylated and all these are negatively charged. So they stay here, and then use your antibody to show it. But when you have an N-glycosylation disorder, whoop, something happened to my slide. No, never mind. This should be gone. So imagine that this is gone. Then you have some of the antennas not so negatively charged. So you will have some other bands which would wander further along. And if there is a type 2, a no glycosylation disorder, you would have even more bands. So this is in reality when you're doing your transferrin isoelectric focusing. This is the control. This is the patient. And now I can go into it. Ha! See, it's gone. And when it's gone, you will get another pattern. So this is the easiest test ever because this is a screening. You can screen all different glycosylation disorders just using this test. Screening for negatively charged sialic acid missing wanted guys. I have to talk to the microphone. Can we go on? I will surprise you with something more exciting. When you are driving and drinking, you never do that. When you're drinking and somebody tests you, maybe takes a drop of blood and tests you for alcohol consumption, they're using this test because alcohol ingestion leads to a secondary glycosylation disorder. So when you're really high, then you would see your own test showing CDG syndrome, and that's actually the test that the cops are using when they are testing us. Not only alcohol ingestion alters your transferrin isoelectric pattern, also two other very common metabolic disorders, galactosemia and fructosemia.
Does that mean that a person with a CDG could be wrongly incarcerated by police?
Absolutely. Although I don't see many patients, driving around. But I have a horror story.
But your patient you described earlier, she could be driving.
What was the question?
The question was that if we can wrongfully accuse a CDG patient of alcoholic ingestion. How you call it, DUI or
DUI?
DUI, yeah. It's always in People magazine and stuff like that. But I can tell you a horrible story. I still cannot believe it that it happened. We had a patient at the wards. I'm working in the Netherlands in a hospital which is a little bit comparable to Tulane, maybe a little bit smaller. We had a patient who was seizing and who came 3 or 4 times in coma and was admitted, and we couldn't figure out what's wrong with him. But when you have such a history, like altered consciousness and, strange story, you always thinking maybe it's a metabolic disorder, an inborn error, something like high ammonia or lactate or whatever. So we did the metabolic screening. And the patient also had epilepsy and was not thriving well. So we were thinking we should screen for CDG syndrome. And it was positive. The baby was 10 months old. And so we almost got into this discussion the the bad news, how you call it, you know, when you're telling the parents the bad news, when you tell them the diagnosis. When the nurse came and she said, I think father is doing something with this child. And he was feeding the child with alcohol. He was poisoning him because he was not normal. And he was poisoning this child with alcohol. So every time he came for the admission, he was just in an alcoholic state. But he really had the classical pattern of CDG syndrome. So we were not, you know, if the nurse didn't see it, we could have, you know, missed this case of abuse. It's a sad story. Anyway, I want to talk about better stories than I have to stay here. Can I look behind me? Can you get the next slide, please? So back to the screening. Yes, DC.
So when you do by tandem aspect, do you still see the same pattern between focusing on CDGs?
So the question was, if we do tandem assay, if we can diagnose CDG, and if we can say whether it's primary or secondary glycosylation disorder. You have to perform a very specific mass spectrometry to find this disorder. Sugars in general are really difficult molecules to see through mass spectroscopy. So we are trying to deal with that, and we have some results, and sometimes you can make the diagnosis through mass spectroscopy, but this is only for the very difficult cases when you're not coming out with your simple screening test. So we are still doing. You are not happy with my answer.
No, I'm just curious because we got a lecture from someone from Greenwood Genetics. But they're using not standard tandem aspect.
Yeah. So if I have to repeat the question, he said he heard the lecture. Somebody at Greenwood is using mass spectroscopy to analyze sugar antennas. It's possible. It's a very delicate measurement. We use it when we are really looking for the fine-tuning of the diagnosis, but it's not a screening thing. So if you just if you have it in your mind, maybe this is a CDG, you should just do the most simple test, and this is it. Okay, and if you want to carry on and really confirm the diagnosis, of course, you have to go for the enzymes, and you saw all these different enzymes, so it's not easy to measure them one by one. There is a most common type. I showed you the phenotype previously. And that's the so-called BMM2 enzyme. This is the most common type of alteration. Or you can analyze the so-called lipid-linked oligosaccharides. These are lipids which are carrying these antennas to the right place. Or you can use the tandem MS if you're really good, or if you're working at Greenwood or in Nijmegen. And you can do your genetic tests. So we are not so smart, I have to tell you. And sometimes we just don't find a diagnosis. We know it's CDG, but we don't find the subtype, so we call that CDGX. It's almost like Mr. X. It's mysterious, but it's very bad for the parents because they really want to know what's going on.
So that's not CDG10, that's CDGX?
Yeah.
Oh, it's a bad nomenclature.
It's a bad nomenclature. Maybe we should just write it up and figure out something else, something better. So there are several labs in Europe dealing with this question and using the whole 9 yards to get to the answer. And there is a laboratory, Hudson Freeze has a very sophisticated laboratory dealing with unsolved CDG cases here in California. And of course, you're the future, so maybe you should solve the unsolved cases. But how would we start? how would we approach these patients? We don't have 10 MMS or like an antennas here at our hands, and maybe we can measure some enzymes, but surely not 36. So how can we solve the unsolved? Actually, most of the end glycan disorders are solved. Almost all the patients. So where we are having a real difficulty is O-glycosylation. So you see this O-glycosylation, this is the control. Remember the abnormal type. When you have hypocellulation, then you get the spread out bands. And you see that the antennas are looking different as well. It's not like missing antennas, but the structure of the antenna is not right. Look at the numbers. We have actually so-called Euroglycanet. We were trying to put our minds and hands and samples together and collected the patients from the Benelux countries, so also from Belgium and Luxembourg, and some of them from Germany. And if you look at the last 2 years, we had 53 type 1 patients, which are all solved, and only 10 are unsolved. But when you look at the type 2 patients, then you have only 4 solved and 21 unsolved. So this Golgi-related glycosylation is still a challenge. It's still a mystery. So I will ask you to help me to solve a couple of patients right now. But first you have to understand how it's going. So this is our screening thing. This is our patient. This is the possible structure of the antenna. We only know that it's not negatively charged. But what is the structure? Is this like this or this or this or this? You cannot really judge from a screening test. Is it right? Because you only screen for missing negative charges, but you're not really telling what's hanging on the antenna still. Next slide, please. So what you really want to know is the structure. Next slide, please. So what we started to do is developing a new test for O-glycosylation. So let's find a new protein which is only O-glycosylated. Remember, so with the hydroxy group hanging on a serine or threonine. This is very easy. Apolipoprotein C3 is such a protein. It's in the mucus. It's a systemic protein. And when you do isoelectric focusing of the antenna, you see a little bit similar pattern to what we do with the transferrin. But as you see, normally you have 2 antennas or 2 branches of the antenna and some with 1 branch, so it's in a build-up phase. And of course you always see some premature forms which are out there and not completely finished. But actually when you do this test, you know what your antenna look like. So we published this test and then we started to screen our patients, all the unsolved, with this new test. Let's think about it. So we have abnormal transferrin. It can be type 1 and type 2. We know that. It's N or O. So let's test the remaining patients with this new test. Yes, please. So this is the classical normal tetracielo form, and this is for the O-glycosylation, the normal distribution of the 2-branched and 1-branched antennas. So when we screened our patients, the remaining patients, we could make 2 groups. One group was totally hypocellulated. Look at how high the 0 form went up. And there is almost no 2-branched antenna form. So this is like a systemic severe underglycosylation. And there is another form where you see there's still some double-branched type, and although you see some elevation of the hypoglycosylated form, the most abundant antenna form is the one-branched. So we don't know what this is, but we see that within our unsolved mysterious group, we have two subgroups. Can we solve this now? This is all right. So we publish that. Every time I'm showing something, then I'm emphasizing the importance of ourselves. So we publish that again. But then this Hudson Freeze guy, who's even smarter than we are, he sort of discovered the first O-glycosylation disorder, which is a combined N and O-glycosylation disorder. So there is a total chaos in the glycosylation. It goes wrong in the first phase, in the endoplasmatic reticulum, and then it's not enough. After that, in the processing and the transport phase, it goes wrong again, so the antennas are not writing right, and you get like both glycosylation wrong. This is a patient which was diagnosed with a syndrome because it didn't really look like CDG. Actually, was a very dysmorphic patient which had fatal liver disease, fatty liver abnormalities. Didn't grow at all. So he was born, and at 6 months old, he had the same weight and length what he had at birth. He had seizures and skeletal abnormalities, and when a second child was born from the same family, it was the same phenotype. So poor parents had two very severely abnormal children. Next slide, please. When we try to give a syndrome name to these patients, we call it microcephaly, adducted thumbs, gross retardation syndrome. The patients had this pattern, a type 2 pattern, with all these little bands, and a profound hypoglycosylation. All glycosylation went wrong. If you look at the syndrome, then you see some features, and these are other patients we discovered a little bit later, a couple of months later at our own wards. If you look at the patients, you see the severe microcephaly. You see some dysmorphic features, very short nose and long philtrum. You see the adducted thumbs. This is a sort of arthrogryposis. The patients are born with their thumbs like this, and they cannot really move their thumbs. But if you look at their position, you see that they have contractures also in the knees and in the hips as well. So this is a particular phenotype. You would say, this I can remember and I can recognize when I see it. And just not only the phenotype, but also a biochemical phenotype, because hypocyanulation. The patient has a so-called Koch defect. And this is an evolving new biochemical group of diagnosis leading to different Golgi syndrome, Golgi diseases, Golgi disorders. So what's the Koch complex? Did you learn anything about the Koch complex and the role of Koch complex in the Golgi? You are looking with a blank stare. Yeah, this is quite new. It's like a couple of years ago they were discovered, and we are still not completely sure what they are doing. These are complexes formed from two lobes, lobe A and lobe B, and they have also 4 subcomplexes, and they sort of work together and have a major role in glycan processing and antenna ripening in the Golgi. So if they're not working, the Golgi will go wrong. If you do like a histology, you will see the Golgi really like changing its form and getting crystalline inclusions, puffy and abnormal, like the structure is completely abnormal. And you would have a completely misplaced antenna, which wouldn't be at the right place at the right time. So this very smart guy, Hudson Fries, discovered that there is a mutation coding for one of the COG complex proteins. We call it COG7. And when you have a mutation here, then these two lobes cannot really get together and cannot form a super complex, and all that glycosylation would go wrong. Next. All the patients had the same mutation. The first two I showed you, which he solved by biochemical and genetic approach. And the other three patients we found in Nijmegen, in the Netherlands and in Belgium, which are completely the same. They look like the same, they behave like the same, they are as severe as the other, like carbon copies, and they carry the same mutations and carrying the same biochemical defect. Even more disturbing, they do have the same haplotype. Did you learn about haplotype? So actually your genetic background, your small details, your background, where did you come from? I'm Hungarian, for example, so probably if you just compare my DNA to other Hungarians, then you would see a lot of similarities when you would do fine-tuning or marker analysis. So sometimes when you try to analyze a pedigree, you do haplotype analysis to see where that individual has come from. So all these patients had completely identical haplotype. The two patients, which was, discovered by Hudson Fries group, and the Dutch patient, and the Belgian patient. So it has to be a common mutation somewhere, and probably these patients are spread out in Europe now, carrying the same genetic background. So this is a syndrome, a specific syndrome. We see it always the same way: hypotonia, adoptic thumbs, heart defect, wrinkled skin. They have wrinkled skin on their neck. Profound feeding difficulties, microcephaly, and failure to thrive. They don't grow. So when you see a patient like that, you would just say, this is it. You can test for the gene. You don't even have to do all that hula with the transferrin and whatever analysis. Okay, so we did solve one. Actually, we didn't solve it because Hudson Fries solved it. But after that, we defined the syndrome, So we are smart, and we have self-confidence. From now on, we will solve the O-glycosylation disorders. What's left? So let me show you an absolutely intriguing syndrome, Cutis-Laxus syndrome. It's intriguing because I don't want to get old. When I see the skin, I'm thinking, someday I'll probably look like this. Maybe, no, I don't even want to go into that. It is a developmental disorder. These patients look like this when they are born. Their skin is old. It's not ripening right, so the elastin fibers, which should be elastic, they are really sagging and not formed right. And so the poor patients have these hanging skin all over their body. This is a patient from Germany, and you see this very peculiar pattern of aged skin on the belly. her skin on the face is not very particular, but she has a very peculiar face. So maybe I ask Chris, how would you describe her face? What do you think what's peculiar?
Well, she definitely it looks almost like pronaphia there, at least the appearance of it.
So Chris says she has prognathia. Anybody else? Something about the face?
There's a prominence of the mid-face too. The cheeks are kind of, I don't know if it's because the rest is sunk in, but it's definitely sort of sticking out more.
So
Some people fissures.
Is that the
It
normal?
doesn't close the mouth.
It's almost a hypotonic. See, like a congenital myotonic face.
So this is pointing out that the mouth is open, so it looks like a myopathic face.
Is it always open? Like not just when you have the picture stage, you could really close it?
I think it's open. She has some muscle issues as well. So prognathia, yeah?
Eyelids. No, eyelids.
What is peculiar about the eyelids?
Down-slanting palpebral fissures. epicanthic
Hans
folds.
says down-slanting palpebral fissures, epicanthic folds.
They actually look short too.
They look short. Do you all agree?
Low light nasal bridge. There's a
Lower.
But I'm
Filtrum is flat
flat, long filtrum.
and micropnasia.
Micrognathia, retognathia. So when we try to describe it, it's not so easy because it's not so absurdly dysmorphic. But we see many minor anomalies, many little things which are not right. And I think that what I recognize in this syndrome, and you will see more patients, so it will be easier to discover the similarities, is the down-slanting palpebral fissures and somewhat shorter nose, long filtrum, flat filtrum, and somewhat prominent ears. It's not so spectacular here. But, of course, open mouths that we see many times, and the sutures are open. So we will see more faces, and then I point out to you what I still find peculiar in their physical aspect. But we see something very strange as well. Look at the fat distribution. The buttocks are almost hanging. See? And look at the arms. I mean, this is the same abnormal fat distribution what we saw with the end glycosylation disorders. So this should be a difficult one to solve, huh? Next slide, please. Here's the pattern. You are experts on transferrin and ApoC3 pattern. You see it's not totally hypoglycosylated, but the one branch antenna is dominating the picture. I show you more patients. This is a patient not easy to see, but same down-slanting features and long filtrum. You see that patient with a very severe cutis laxa. You see that fat distribution abnormality. This patient is already 14, so you can see how the fat is developing. And cutis laxa on the legs and on the belly. These are the clinical features of this new cutis laxa syndrome. They all have cutis laxa, so sagging, hanging, old skin. They're hypotonic. So they have a hypotonic face, but also hypotonic muscles. They have very lax joints. They have all these subluxations. They have microcephaly, and that's very strange because their fontanelle stays open till the age of 3, 4. Large, huge fontanelle, but their brain's not growing. And some of them have deafness or seizures. It's very interesting how the phenotype develops. Because look at her, how much excess skin she had at the beginning. And she appears less wrinkly a little bit later on. And when she's standing, she's holding her skin. But actually here, you don't see much cutis laxa at all. So it's not even variability in between patients, but variability throughout life when you see that the major features are vanishing in the time. But of course you see the features again, the downslanting palpebral features, open mouth, the short nose, philtrum, and so. So I think they she reminds me, she's a German patient again, but she reminds me of the other one. To surprise you more, this is also a brain developmental abnormality. Have you heard about pachygyria? We have normally, when we are normal, we have gyration on our brain. And that's how we can think. If we have no gyration, then we fall into sleep. And then we keep on sleeping all our lives. We cannot really think well. So what you see here is pachygyria. This patient has almost no gyration on its brain. And this patient is less severe, has a partial pachygyria. Here it's normally gyrated, see? And there's one large block where no gyration is to be seen. But we know this syndrome. We know this pachygyria. There are other syndromes with the same thing. Have you ever heard about muscle-eye brain disease or Volcker-Warburg syndrome? These are major developmental disorder syndromes with abnormal brain and abnormal muscle. And I have to tell you, these are also glycosylation disorders. So I'm not surprised that this patient has the same problems. Okay, let me cheer you up with the fact that we have about 5 minutes left, and then we are done. I want to show you how did we solve this syndrome. Actually, we solved it based on the fact that almost all patients we saw came from a consanguineous family. So we were thinking, if somebody is accidentally carrying a recessive mutation and would marry someone from the same family, has a higher risk to get a disorder than another one who's marrying somebody from another family. So if we use this to solve this problem, next slide please, then we have to screen all the chromosomes, all the genetic material of these individuals and look for same genetic background. So we were not thinking, we were just screening. We screened chromosome 1, 2, 3, 4, all the genetic material, and we only screened for places in the genetic material where father and mother had the same information. We didn't know which gene we should look for. We only knew we are looking for same background, same haplotype. Next one. So that's how we went. These were the first three patients. This is a German patient. This is a patient from Turkey, and she's living in the Netherlands. And this is a third patient from Turkey. Next one. And what we found, we did found a region on chromosome 12 where all these three patients had identical genetic information, totally identical. Of course, there could be a crossing over sometimes, which would disturb our information, but this was just pure luck. This was completely identical. So that was, at the first time, it was too large to really go into genes and look for genetic background. But then we collected patients from all over Europe, and actually also from the States. And finally, we end up with 12 families. And already, when we reached 7 families, we had a common region which was not so absurdly large that we couldn't have looked for a specific gene. We were extremely lucky. Because this ATPase gene is a known gene, which has a brother, and the brother is known to be causative in X-linked cutis luxus syndrome. So when you're looking for a gene to sequence to find the disease, and you're always thinking, how should I look? Maybe I should look into elastin-related genes, or brain-related genes, or glycosylation-related genes, but we didn't find anything in this region. But this ATPase gene, it's an ATPase6 gene, is very similar to the ATPase7 gene, which is causing the X-linked cutis luxus syndrome and also Menkes syndrome. So we thought, there's the money, and we went to sequence it. And we really did find a mutation there. And we found several different mutations, and this is that ATPase gene. And I will surprise you with something. This is a gene which is responsible for the proton gradient. So the proton gradient is important within the Golgi, because when you try to get those antennas throughout the Golgi from one place to the other, you use your proton gradient at the two sides to have those antennas go along and go through the certain steps of the Golgi. So if an ATPase enzyme is not working right, those antennas will get stuck somewhere in the middle of the Golgi and would never get to the right place. So this is a completely different concept. There was nothing wrong with glycosylation. It was all coded right. Only the stupid enzyme, which would help the antennas to go to the right place and set on the proper protein at the right time, it was not functioning right. So they just got stuck in the Golgi and never got to the right place. So we have many different mutations. The first reports were mostly nonsense mutations, and later on we found distance in different types. We have about 25 new patients since then, and you see some clinical variability there as well. But the amazing concept in glycosylation, that we started out from a genetic concept or for coding for a genetic background, when the glycosylation enzyme is not working right, so you don't get the proper form. But now we know that we can have a normal glycosylation through other problems, through the surrounding proteins and regulations, which are also affecting glycosylation. So actually it's a secondary glycosylation disorder, because it's not coded like an enzyme or like an antenna structure gene, but it's just the surrounding of the process. So now it's really difficult to understand that, but it's a nice concept anyway. If you see a phenotype, it can go wrong because of the structural proteins, that you have a gene coding for a protein, have a mutation, you have a bad protein and doesn't work. Okay. But other times you have that protein completely right, only your glycosylation enzyme is not working, and you can have that protein beautiful, but when the secondary structure is not right, it's not working. But that's not enough. You can have also another enzyme which is only coding for the surrounding of all these processes. So you have everything right, the protein and the secondary structure, only the surrounding is not working, and finally you will end up with the same problem. So it's complicated, but it's challenging, a lot to do. I really appreciate your enthusiasm and your smiling faces to me. It was a pleasure to talk to you. Thank you.
Questions?
Yes.
In the patient, when you had the mutation in the hydrogen ATPase, was it just the mitochondrial ATPase, or was it ATPase in the kidney, for example, or other organs?
The question was if this ATPase is identical with the mitochondrial ATPase. No, this is
No, was there a mutation in other ATPases, or is it that ATPase that?
It's not the mitochondrial ATPase. It's a V-ATPase. So the structure is almost completely identical with the mitochondrial ATPase, but this is an ATPase expressed in lysosomes and endosomes. So a sister of the mitochondrial ATPase.
One more question. Are lectins useful at all for checking any of those diseases?
I love this question. She's asking if we are using lectins to do this barcoding evaluation. A brilliant idea. Lectins are something like antibodies, you know, they are sort of recognizing glycan antennas. It's wonderful if you have that in hand. It's one of the best diagnostic possibilities for glycosylation. We only use it in muscle disorders so far, not in systemic glycosylation, but that's a brilliant approach to solve the unsolved.
Okay, well,
My
thank you.
question is that what is the difference between this and Michelin tires?
The question is if there is any relation to Michelin syndrome, Michelin baby. Do you know that Michelin baby syndrome? You know what the Michelin baby is? The tire baby, you know, sometimes you see those advertisements with Michelin tires. It is an unsolved genetic syndrome which has several overlapping phenotypic features with this, because it has also abnormal fat distribution, but in general they don't have cutis laxa. So when you biopsy their skin, they have normal elastin structure, but the abnormal fat distribution they do have, and maybe it's related to glycosylation.
Any other questions?
See?
Great.